For 75 years, Argonne National Laboratory has accelerated the science and technology that drive U.S. security and prosperity. To celebrate, we're capturing the stories of the people who've made it happen. This is Argonne Voices. In response to the COVID-19 pandemic in 2020, scientists produced effective vaccines in record time, thanks in part to research conducted nearly a decade ago at Argonne's Advanced Photon Source, or APS. The APS works like a giant X-ray microscope. The ultrabright light allows scientists to see deep within materials to understand their makeup. Jason McLellan, a structural biologist, has conducted research at the APS for years, studying the protein structures of different viruses. In conversation with Bob Fischetti, life sciences advisor at the APS, Jason explains the nature of his research that laid the groundwork for the development of life-saving COVID-19 vaccines.
Hi, I'm Bob Fischetti. I have two roles at the Advanced Photon Source at Argonne National Laboratory. I'm the Life Sciences Advisor to the APS Director, and I'm also a group leader for GMCA, which is a group that's funded by NIH to build and operate what we call X-ray beamlines for structural biology users to come in and look at diffraction data, to look at the structure of proteins. I'm here today to talk to Jason McLellan, who's been doing a lot of research at the APS and has been heavily involved in some of the vaccines that are now in my arm. So, Jason?
I'm Jason McLellan. I'm trained as a structural biologist, and we try to determine structures of viral proteins, their complexes with receptors and antibodies, and use that information to engineer vaccine antigens.
So part of my role at the APS as the Life Sciences Advisor is to help publicize some of the research that's done there for drug development and for various other things that are important for human health. And recently, there's been a lot of interest in COVID-19 and the work that's been done there. And to a lot of people, it seemed like this vaccine just appeared overnight. There was actually a decade of research that went into this. So maybe you could highlight a little bit of some of the really important steps over the last 10 years that made it that you could so quickly bring out this critical vaccine.
Yeah, it's a great point. The vaccines and components of the vaccines have been in development for over a decade. If we think about the two mRNA vaccines, the one from Moderna and the one from BioNTech and Pfizer, they can be thought of as having two components. So one is the mRNA delivery platform. And that has been studied for over a decade and longer about how to make the mRNA, what sorts of modifications to include. And the mRNA is essentially like injecting a recipe into our cells and instructing them to make a foreign protein from the virus. So that way our immune system recognizes that foreign protein and develops a response to it as if we had been infected. And so although these are the first mRNA vaccines that have been approved for emergency use authorization, many of them have already been in phase 1 and phase 2 clinical trials. So even though it's new, it's a relatively mature platform, and its major advantage is speed. It doesn't matter what the mRNA is encoding, what the recipe is, you make it the same way. So it's really great in responding quickly to a pandemic. And then the other component of the vaccines is the actual antigen, the portion of the virus that the mRNA is encoding for, what the instructions are telling our cells to make. And that's where our work has come in. So I first started working on coronaviruses back in 2013 in collaboration with Dr. Barney Graham at the Vaccine Research Center at the National Institutes of Health. That was around the time that the Middle East respiratory syndrome coronavirus had emerged in Saudi Arabia and the neighboring countries. And that was the second coronavirus to jump species to enter the human population in the last 10 years. So we had the first SARS outbreak back in 2002. Then we had MERS at the end of 2012, 2013. And we thought, well, we've just had two separated by 10 years. There's likely going to be another one 10 years from now. So we should start trying to figure out how to respond to coronavirus outbreaks and determine the best possible way to make a vaccine. And at the end of 2019, we were all, I think, in the scientific community, and the virologists anyway, were following these reports of pneumonia outbreaks in Wuhan, China. And we were, of course, very nervous about that and thought, well, it could be a new flu outbreak, possibly a coronavirus outbreak. And then I think it was confirmed January 3rd that it was, in fact, a beta coronavirus, similar to the first SARS.
One of the things that I think is interesting about that story is that Barney Graham called you and you were on vacation with your family on a ski trip and you're on the mountain and he calls you and asks, do you want to work on this?
Yeah, that was January 6th. So it was about 4 days before the genome sequence was released. And we were out snowboarding in Park City, Utah. And I thought Barney was just calling to wish a Merry Christmas and Happy Holidays. And he let me know that the pathogen that was causing these pneumonia outbreaks in Wuhan, China, was a beta coronavirus. And they wanted to rush and make a vaccine, wanted to know if my lab was interested in helping, designing the spike, determining the structure of the spike protein. The spike protein is this very large club-like or mushroom-like molecule on the surface of the virus. And the spike protein binds to specific receptors on our cells. So it attaches the virus to our cells. If we think of it as a mushroom, the cap of the mushroom falls off, the stalk extends and shoots part of itself into the host cell, into our membranes. And then the cell membrane and the membrane of the virus fuse and allow the genome of the virus to enter the cell and take over the cell. And so what we've been studying since 2013 is trying to understand the structure and the function of the spike protein. And so that's what we helped determine the first structure of a spike protein from a human coronavirus back in 2016. Then my lab used that spike structure to make small tweaks, like adding little staples to help rigidify the protein. And we showed that that's a better vaccine antigen when it's stabilized. And we had all of that work done back in 2016. We knew how to express the spikes, how to add the stabilizing mutations. And so as soon as we got the genome sequence of the new coronavirus in early 2020, within hours, we had designed a stabilized spike protein. Moderna was already starting to make the mRNA that could code for the stabilized spike protein. And I think within 65 days, so we went from a genome sequence to the first injections into humans, which is really remarkable.
The advanced photon source is actually a large particle accelerator. We can computationally go back and determine the structure of the proteins. We can see where all of the atoms are in the protein. And so work for, like Jason does, you want to know, here's an antibody. How is it going to bind to the spike protein? And then he will take that and work with others and they'll develop something that's even better. And so I think that's what you've been doing for sort of the last 10 years using the facilities we have available at Argonne.
That's right. Initially for a lot of these pathogens, these viruses, we have sort of a very low resolution, almost cartoonish view of what the proteins are and what they look like. But what we want to know is exactly what they look like to make a very detailed image where we know the location of every atom in these proteins. And by doing that, then we can start to turn it from a guessing game into a very rational engineering approach to modifying these proteins.
I mean, it's exciting to see not only the expertise from the local people who are on site, who develop the instruments and operate them, who are also scientific experts, and to be able to see all the experts that come in and to be able to help facilitate people working on all kinds of different problems, biomedical problems, things that at some point are going to make a big difference.
Yeah. So my lab and others, we've used it to determine structures of viral proteins like the coronavirus spike or portions of the coronavirus spike in complex with receptors, in complex with antibodies. And it's really illuminated a lot of biology, understanding how these viruses function, how do they get in. And it's also provided a lot of information to the development of antibodies, small molecules, and vaccines.
So thank you for all the hard work you've done. And it's incredible, the results that it's produced and many lives that have been saved.
Thanks so much.
Argonne Voices is an oral history project, recording the stories behind decades of world-changing science at the laboratory. To learn more about Argonne's 75th anniversary, visit anl.gov.